topic that is very relevant and timely, I guess. So we'll talk about some of the works that we have been doing on COVID-19 diagnostics. And just to give you a brief background, that I recently moved from University of Illinois Urbana-Champaign, and with a dual appointment between engineering and medicine. And I'm affiliated with radiology, pediatrics, biochemical, and environmental engineering, and computer science and electrical engineering at University of Maryland, Baltimore. And my disclosure, NIH grants, founders of startups, and with that, so COVID-19 global pandemic, I mean, that kind of changed the way we lead our life. And for the last 1 year or a little over 1 year, our everything has changed. The way we lead our life, the way we do our research, the way we operate. The first case was reported in China in December 2019, and by March last year, WHO recognized this as an outbreak, this outbreak as a pandemic. So far, 124 million worldwide confirmed cases as of today, and more than 30 million cases in the US. 2.74 million deaths worldwide, and just in the US alone, we have more than 500,000 deaths. So at this point, there is a real need for global mass testing, and with the demand that we can estimate to be more than 600 million. So that's and that will include the genetic test and 500 million of rapid tests. The genetic makeup of SARS-CoV-2, the virus that is the reason for COVID-19, is pretty interesting. It is a tiny virus, only 30K base pairs, and the genome has the nucleotide identity of 96% with the bat COVID virus or coronavirus, 91% similarity with pangolin coronavirus, and only 80% with the SARS virus that was present a few years back, and 55% with MERS, and the common cold flu virus only 50% around. The SARS-CoV-2 spike protein that is present on the surface of these viruses, they're kind of optimized for binding to human-like ACE2 receptors, and that is the result of a natural selection. Now, the mutation happens to the spike protein, and although the mutation is not so prevalent, but it does happen, and as a result of that, the SARS-CoV-2, that allows the virus to partially escape from vaccines or existing herd immunity, and that makes it very difficult for getting accurate tests that kind of target the antigen response or based on the antibody that are present. So the global market that we find today is more than 16 billion opportunity for diagnostic tests, and by 2025, and that's kind of predicted. The focus customer, as we know, that the need is really need of the moment is community testing sites, schools, airports, churches, you name it. And most importantly, low and moderate complexity laboratories that can be certified by CLIA, that is Clinical Laboratory Improvement Amendments, and point of care testing by facilities operating under CLIA certificate waiver. That means the tests that can be run by less trained or not really expert and skilled personnel. Those tests are of really of high importance these days. The performance criteria for various analytical approaches for diagnostic for viruses that we can find is that if you look at it, then the gold standard is really the RT-PCR, and that's really a big device. And as we know that the large laboratories and hospitals, they would have RT-PCR that provides a sensitivity of around 97% with a specificity of 100%, and the limit of detection would be somewhere around 100 to 1,000 copies per milliliter. And the test time is typically 2 hours, 2 to 3 hours, but because of the rush, because of the number of samples, and because we are going to require skilled personnel, typically the turnaround time, the sample to answer time, is 4 to 6 days. In today's time in the US, we're looking at around 2 days turnaround time. The price price for the platform is somewhere between 15,000 to 90,000, and the technological maturity would be very mature. In comparison to that, the point of care devices or lateral flow devices like the pregnancy kit type things, those are really emerging, and they don't really, at this point, there's a very little information out there. There's very little low number of tests out there that are FDA approved or that can really bring the same advantage that the RT-PCR can bring, but with a very short turnaround time and keeping the price low. So the entire goal for this research is really try to come up with some point of care approaches, the molecular diagnostic approaches that can produce results comparable to RT-PCR, but keeping the cost low and with a rapid result, and where we're not going to require any skilled personnel to run these tests. Now, if we look at a characteristic infection progression in a single patient, then you will see that the virus has an infection period or incubation period for 5 days. So for 5 days, the person has been exposed, and the latent period would stay there for 3 days. That means for 3 days, the patient remains completely asymptomatic, or there is no symptoms shown. After that period, there is a possibility that the person remains either asymptomatic or start showing symptoms. So the diagnosis after 5 days is presumably too late, because at that point the person is showing symptoms and highly infectious. So the goal here really is to identify the infection at a very early stage, and possibly within this latent period of 3 days, where if we can detect the virus at that early stage, then there will be we can really restrict and minimize the infection and the spreading, and also the treatment can be applied pretty quickly. So with that goal, the point of care test for SARS-CoV-2, where we really target because we know that the early detection is the key. We target RNA-based detection, and I'm going to explain in a minute. We target inexpensive test, limited resource use, easy to operate, highly sensitive, and we will have the lowest possible detection range. And also we should have high specificity for the virus, and should be able to differentiate between other coronaviruses and other related viruses. So this is a plot that shows the expression of the virus and the production of the antibody over time. And as you can see here, that the red plot, that kind of shows that we're at 3 days, that starts the presence of antibody or IgG or even IgM that starts producing in the serum of the person. So that means the person is really, the body is really responding to the presence of this virus from third day, and then it picks around at 14 days. So almost at 14 days is no detectable SARS-CoV-2 viral RNA, but there will be an antibody present. So if we test the blood of that person, we will see the antibody is present there. So at that point of 14 days, the antibody tests are useless because we were not really detecting early enough. We really need to detect somewhere from the 0 day to third day where the person is not really producing antibody, but the RNA of the virus is really present there. So the importance of a molecular test is absolutely undeniable. So knowing the enemy, the viruses we were talking about, that the spike trimer, as you can find here, that this is the protein that is responsible for binding to the SEE2 receptors on a host cell, and it is also primed by another protein that is TMPRSS2. Now, compared to that, the N protein or the N gene that is inside the virus, or nucleotide, that is less prone to mutations. So as a result of that, it is absolutely understandable that targeting an targeting an N protein or N gene would be nucleoprotein would be much more safer and for a test. So our goal was really to go after N gene for that particular particular virus. So there are two approaches that we're following. The approach one is a plasmonic detection. This is the process flow where the patient can or the subject can really get a nasal swab and then get the sample, and then the sample pre-preparation. The RNA can be isolated, and it will be incubated at 65 degrees for 30 minutes for nucleic acid amplification, and then the colorimetric detection can happen where the positive will be kind of bluish color, and the negative will still remain as a purple or kind of reddish. Now, so the whole idea is really we're basing on antisense oligonucleotides that are we have developed antisense oligonucleotides that are targeting the N gene of the SARS-CoV-2. For the detection, the plasmonic detection is the resultant of using the highly precise gold nanoparticle of 20 nanometer in size. The idea is that the gold nanoparticles that are capped with these oligonucleotides, when in presence of the viral RNA, they will form the agglomerates. They will precipitate. They will form large clusters. And because gold particles are plasmons, plasmonic response based on their size and the assembly, there will be a change in the color, and that change in the color can be appreciated by a selective naked eye detection. So very simplistic, inexpensive. The result can be obtained in 10 minutes. It can be made quantitative if connected to the spectrophotometer, and the detection sensitivity can reach around 10 copies per microliter with a specificity of 100% and sensitivity of 98.3%. And a very little sample is required at 3 to 5 microliter of RNA. Can be integrated with a lateral flow assay where you can produce results as like a pregnancy kit, and also potentially be tagged with a color sensing mobile app. So the design principle of the antisense oligonucleotides targeting N gene, and this part of the work was done in my lab by Dr. Parikit Moitra, who is a junior faculty in my lab. So what we're doing here is a 4-point binding. So this is a claw approach where we're looking at the front end and the end location of the N gene, and we are now designing 4 different oligonucleotides. So 2 of them bind to the front location and 2 of them bind to the end location. So the design principle in such a way that it's a tight binding, and the moment these gold nanoparticles, they come in contact with the RNA, they will form an agglomerated gold particle, and as a result of that, there will be the plasmonic response. So as you can see here that this is a perfectly separated, well discriminated gold particles when they're ASO capped, but in presence of the N gene or the RNA, there is a red shift in the absorbance and the 40 nanometer red shift, and that results in a cluster of gold particles, and as a result of that, there is a change in the color. So you can see here that these are nasal swab samples collected from COVID positive and negative patients, and you can see here that the positive samples, they turned blue, whereas the negative samples, they remained red. And we have done hundreds of tests with clinical samples to confirm that this is the case. Same here, we can also use saliva, and what we have done is that we have spiked saliva with RNA, and we are showing that the same result can be obtained with saliva. And also we are showing here that the selectivity of the test that marks COVID RNA that did not change the color. As a result of that, we can conclude that this is a highly specific detection process. So we've also done a direct testing so that this becomes a real, in a true sense, point of care, where the samples can be directly collected from the clinical samples collected, and then no requirement for RNA extraction. We can apply some lysis medium, and so the sample can be mixed with the lysis medium, and then directly be included with the combined with the reaction mixture. And what we are seeing here is that the result is fantastic, and we can really confirm that the positive samples and negative samples, we can really isolate them pretty well. When we published this work last year in SCS Nano. But the question is, so with 10 copies per microliter, the detection sensitivity, so we really need to, if we look at this, and this is a plot from a recent paper that that was published, and what we are seeing here is that the fraction of cases on the y-axis, and then x-axis, this is log 10 RNA, or the genome copies per ml. So what we are seeing, this is from Journal of Clinical Virology, from other, this is not our work, but from literature. So our plasmonic test really can detect everything that comes within this red box. This is our target population. But what about these the population that is below that? So log 10.1 to log 10 3.5. So that's a good question, and I think that we can approach this thing slightly differently. And instead of applying a plasmonic approach, we can probably apply an electrochemical approach to produce a much more sensitive result. So also this plot, the second plot, that shows the fraction of cases detected with genome copies. You see that our POC plasmonic test is right there, and all the other tests that are FDA approved, like SOPHIA 2 antigen test or the other tests, they are below our sensitivity limit. All the other tests that have higher sensitivity, they're all with the RT-PCR test, which is not really a point of care test. So if we look at that, so this is the approach 2. This is the electrochemical detection, where the real-time detection is possible. This is less than 5 minutes for stable response, highly quantitative, and we can reach 6.9 copies per microliter. No amplification is required. And so what we're doing here is that we're using a graphene-based platform. So this is a graphene-based platform, and we have a contour electrode, and we have the gold oligonucleotides that were developed in previously for the plasmonic work that deposited on the filter paper. So, and then the hypothesis is that the moment there will be the viral RNA binding, then there will be an upward pull, and as a result of that, because of that upward pull, there will be the change and shift in the electron movement, and that will be detected by the impedance measurement. We also published this work in 2020 last year in a separate SCS Nano paper. So if we combine now the good thing is that if we combine an amplification step, then the sensitivity can be achieved very high, so 7 copies per liter. So this is a little bit of data that shows that this is the ACM image of the sensor that we have developed. And this part of the work was done by the graduate student in my lab, Maha Lafif, and the research specialist, Ketan Dige. And this whole thing is based on paper-based. And here you're seeing the electrochemical response of the device, where the addition of the SARS-CoV-2 viral RNA leads to a spike in the voltage, and then it reaches within 160 seconds or 180 seconds, it kind of reaches a stable result. You can see that how you can separate it from MERS-CoV and the SARS-CoV. Clearly, the SARS-CoV provides a very high the difference in the voltage as the RNA binds to the sensor. Again, we have tested it with a large number of clinical samples. As you can see here, that the positive COVID-19 samples showing the blue curve, whereas the normal sample is absolutely no change in their resistance. So this has been confirmed by many clinical samples. As you can see here, that the positive sample, 59 positive samples were tested, and 30 negative samples were tested, and the confusion matrix showing that we have reached accuracy of 100% with a sensitivity of 100% and a specificity of 100%. So this was outstanding result. So what we have shown here is that we developed 2 assays where both of them are based on highly specific antisense oligos for N gene, and we were applying the nanoparticle platform, which is a gold nanoparticle. Now we have 2 detection capabilities. One is the visual or plasmonic, which can be done in minutes, 10 to 20 minutes. You can really get the whole result done. And the detection limit is a nanogram per milliliter. With the electrochemical, that can be reached in up to femtogram per mil, and the result can be obtained. The assay response time is literally in minutes, less than 2 minutes you can achieve. So with the conclusion, what we are showing here is that we are literally combining molecule making and device making, and an early detection of the SARS-CoV-2 virus is possible at a molecular level. And the translatable solution for COVID-19 early detection has been proposed here. Plasmonic Technologies has already been licensed to RNA disease diagnostics. This company also licensed our electrochemical technique. The FDA approval process is in progress. There is another spin-off company, Vitruvian Bio, that is also developing a slightly different version of the electrochemical test. With that, I'd like to acknowledge the funding from NIBIB. That was both the plasmonic and the electrochemical was funded by NIBIB emergency funds, and other funding also gratefully acknowledged my lab at Illinois and now at Maryland, and all my collaborators. And with that, thank you so much for your time.